Hello. So before starting talking about my project, I'll tell you about PFAS. What does PFAS mean? You can go to the next slide. So PFAS is this group of compounds, more than 10,000 compounds, man-made compounds, that are known to be toxic and really persistent. They share some interesting characteristics. For example, they have, all of them, they have carbon and fluorine in their molecule. I highlighted here PFOA and PFOS because I'll be talking about them a lot in this presentation. And they are well known to be toxic for the human and wildlife. So for I highlighted here some major objectives for our project that are quantify the concentrations and composition of PFAS in Lake Michigan fish, and also to evaluate the dietary routes for PFAS exposure in prey and predator fish with stable isotopes. You can go to the next one. So some exciting finds, but not super happy, is that PFAS was found in all the 9 species that we analyzed. And PFAS, that compound that is known to be toxic, they represent in all of them, all of those 9 species. Yeah, and it's really interesting because this compound, it was phased out from the US more than 20 years ago. So we still have some sources of those compounds, and they are really, really persistent. Something that can make us a bit happier is that PFAS concentrations in Lake Michigan were lower than in other Great Lakes. For example, in Lake Ontario and also in Lake Huron, but still, we still have them here. And the PFAS accumulated differently across salmonid tissues. We are expecting to see more higher concentrations in liver, for example, or also in muscle, but we saw that female eggs has higher concentrations than in other tissues for salmonids. You can go to the next one. So here I brought this figure, as we don't have time to go through the figure in details. I'm going to highlight some things here. I want to call the attention for this dark bar. So before that, in the right figure, you can see the concentration for the 21 PFAS compounds that we analyzed. And in the right figure, you can see the relative composition for those compounds. So here we have the 9 species and divided in predator and prey fish. And this black, it's blue actually, but here you can see almost black. Yeah, thank you. It's the PFOS. So PFOS were found in more than around 50% of the others PFAS analyzed. And you can see here with the composition that the fish, they accumulate different species, they accumulate differently. But I will call attention here for the top bar, that is slime scooping. You can see that you had, in the right figure, you had to put them in a different box because they accumulated almost 10 times more compounds, actually at least 10 times more PFAS than top predators like Chinook salmon and lake trout. This is incredible, and it is mostly related to their diet. And you can see here that deep water slime scooping and deep water scooping, if you look to the right figure, they share similar colors. It means that they share similar source of compounds. It can be related to similar source of compounds, but as slime scooping has way higher concentrations than deep water, something interesting and powerful is happening with the slime scope that we are trying to figure out right now what's happening. Let me see if I can tell you something more about that. Yeah, I think that's it. We can go to the next one. So now for the next steps, now we saw the bioaccumulation of PFAS in all of those species in more than 200 samples, 200 fish. And with the stable isotopes, you're going to see the biomagnification of them. We're going to look to the biomagnification of those compounds. And also, we're going to look to the different species in different locations. We can see it also with the stable isotope, combined stable isotopes and PFAS signature. So we have all the data that you need for those next steps. We are now in the phase of writing the papers and reports. And no, it's not. Because we saw that interesting find that PFAS accumulate different in different tissues. We're gonna in salmonids, We are going now to analyze native fishes and see if it also happens in native fish, also in different trophic levels. So yeah, stay tuned to know more about our research, and thank you so much.